Hey, fellow peptide pathfinders. Rivera here, your chief investigative scribe at Peptides.today. If you're not staying current on dynamic bio-inspired materials, you're missing a massive frontier in research. Today, we're diving headfirst into peptide hydrogels, materials that assemble, dissolve, and even reassemble on command using simple chemistry. Get ready for some mind-bending molecular magic that's reshaping how we think about advanced materials. Let's go! Scientists are now building peptide hydrogel systems that behave like next-generation smart materials in lab settings. These dynamic materials mimic living systems, offering formation, disappearance, and return depending on conditions. A peptide hydrogel is a soft material from amino acid chains self-assembling into water-trapping networks. These gels respond to temperature, pH, or specific chemical signals during laboratory studies. Researchers value peptide hydrogels because they are tunable, biocompatible for specific research, and responsive. Peptide hydrogel systems rely on weak interactions, including hydrogen bonding and beta sheet formation. Individually weak, these interactions combine to build powerful, complex molecular structures without permanent bonds. Scientists call this process self-assembly. Molecules organize themselves into stable, functional materials for research. This self-organization resembles protein folding, a key reason peptide hydrogel research is booming. The hydrogel in this research uses a clever partnership between two distinct molecular components. One component acts as an electron donor, while the other functions as an electron acceptor. When these molecules meet, they form what researchers call a charge transfer complex, enabling interaction. Key molecules include purine lysine cysteine and phenylalanine substituted naphthalene diamide, working in concert. Pi stacking interactions drive their alignment, much like molecular pancakes stacking into organized structures. These columns bundle into fibers, intertwining into a network that traps water, forming the peptide hydrogel. This peptide hydrogel forms rapidly in alkaline conditions, a fascinating initial response for researchers to study. However, the real magic lies not just in formation, but its precisely timed disassembly capabilities. Mind blown yet? We've just scratched the surface of how these peptide hydrogels put on their molecular show, but the plot thickens with their pH clock mechanism and what that means for advanced material systems. Before we dive into those incredible applications, make sure you're following me, Rivera, for all my peptide rabbit hole explorations, and definitely hit follow on Peptides.today for your daily dose of cutting-edge research. You won't want to miss what's next. The heart of this peptide hydrogel system is an innovative chemical pH clock mechanism. Glucono delta lactone, or GDL, slowly changes the solution's acidity over time. GDL converts into gluconic acid, gradually lowering the pH of the experimental solution. Initially alkaline, the hydrogel forms. As pH drops, the gel begins to precisely dissolve. This transformation creates a timed cycle: assembly, stability, then dissolution on a schedule. Scientists tune the gel's duration by adjusting GDL concentration, offering incredible experimental control. Controlling material state on a schedule represents a major breakthrough in material science and research. The peptide hydrogel doesn't just disappear. Researchers can trigger reassembly by restoring alkaline conditions. Repeated assembly and disassembly cycles are rare for synthetic materials, making this highly valuable for studies. This dynamic behavior mirrors how biological systems operate, inspiring new frontiers in material research. One exciting application is advanced material delivery systems, enabling precise release for lab studies. Imagine responsive materials remaining fluid during transport, then gelling under specific lab conditions for controlled release. Smart material surfaces are another promising area for developing responsive interfaces for research tools. In tissue engineering, peptide hydrogels mimic the soft, hydrated environments of natural tissues for cell studies. Cells can grow within these gels, making them promising scaffolds for investigating regeneration in vitro. Supramolecular chemistry plays a central role, allowing precise control over hydrogel properties like strength and responsiveness. Tuning a hydrogel's lifetime is a key advantage, customizing materials for diverse experimental applications. And that's just the beginning of the peptide hydrogel story. These dynamic, responsive materials are redefining possibilities across materials science and fundamental biological research. The ability to program assembly, dissolution, and reassembly on a precise timeline is truly revolutionary for exploring complex systems. The future of programmable materials is looking incredibly bright. Make sure you hit that follow button for me, Kai Rivera, and subscribe to Peptides.today so you don't miss out on the next big discoveries. Before we sign off, a quick legal disclaimer. This podcast is for informational purposes only and is not medical advice. All compounds discussed are for research use only and not for human consumption. Always consult a licensed healthcare provider for medical needs and treatment.